So the good news is there are only 2 slides of notes today. The bad news is it's a fair amount packed into those 2 slides. So keep your game face brain on and stick with me, and we will, I think, finish slightly early. If you are wondering about tomorrow, the time slot we have chosen for the test point recovery thingy is our normal time slot of 11 to noon. Please let me know if you have any other teachers who have anything mandatory for that time slot. We didn't have anyone with conflicts first period, so I'm going to stick with that. But if you've got a teacher that's got something going on, let me know. So there'll be that. And then you have homework you'll have today that is due at the end of tomorrow. There might be a short video from me. I'm not sure yet, but there will be no mandatory meet for this class tomorrow at all. So little break for you. All right. So quick review. Again, make sure you got your paper shapes ready. We'll get to them in a little bit. That gene expression is this process not where the DNA gets copied. That was replication. That was for the purpose of dividing a cell. You want to copy the DNA in that S phase. Gene expression is doing what you're supposed to do with the DNA, which is DNA is a code for protein, right? So 2 parts to it. The first part we will explore today is called transcription. Transcription is where you split the DNA, build a strand of messenger RNA off of it, and then that strand detaches and goes out of the nucleus. That's what we'll explore in detail today and model with our paper shapes. After that comes translation. We'll also model that today, but we won't take notes on that until later. I might have you take a couple of those notes tomorrow, but mostly they'll be Monday. So that is where we're going. Before we do, we need a brief side trip into what genes are actually for. As I mentioned, they are codes for proteins, and that sometimes doesn't sound like enough. So we need to talk real quick about what proteins actually do for you, because it's a lot. You can really truthfully say that directly or indirectly, they're kind of what makes you tick. They're kind of what makes everything in your body happen. In a pretty real sense, a lot of what you are, everything you're doing right now, for example, thinking and taking notes and moving muscles to write and all that, is happening because the right chemical reactions are happening in your body at the right times. Well, the reason that's working all the way it should is a class of proteins called enzymes. You might have heard of them before. Enzymes are the body's catalysts. Catalyst is a chemistry term that means something that basically encourages a chemical reaction to happen without getting used up. And so when living things want chemical reactions to happen, they make specific enzymes for those reactions. And those enzymes happen to be proteins. So every chemical reaction in your body is happening because you're making the right enzymes based on DNA code. Secondly, not only is it making all the right reactions happen, but yeah, you're built from cells. You knew that. But what's keeping those cells together, kind of what, I don't know, maybe the glue that holds everything in your body together, that's all proteins too. They're called structural proteins. To give you an example and bring home how amazing structural proteins are, I'll let you finish writing. Then we're going to do a little experiment with our faces. Okay, ready? I'm going to do an experiment to show off your structural proteins. You ready? Put your hands up like that. Bear with me. Put your hands on your cheeks. Now grab your cheeks and stretch them out that way. Stretch your face as much as you can. I'm sorry if that hurts and you're chapped. But like, okay, you don't have to pull that hard, but like, think about if you really did pull like really hard. Like I can yank pretty hard on my face, which by the way is baby soft and smooth. Very, very, soft, but then I can pull on it and it doesn't tear. Right? There's no chance I'm going to tear my face in half. Sorry, Mikey. Did you get makeup on you? Okay. So there's no chance I'm going to tear my face in half, and yet it's stretched like crazy to where I looked like a weirdo. That is because of a structural protein called collagen. It's what makes your skin so elastic, yet still pliable and soft and stretchy. Collagen and its partner keratin, which you might have heard of. Both of those, oddly enough, wind up on the labels of like bottles of beauty product. They love to put collagen and keratin in things like lotions and, stuff to make you look younger because it's the stuff that your skin's made of. Yeah, putting that in your lotion isn't really going to do anything, but it sounds nice and it sells product. So, but those are just two examples of the many structural proteins that are basically holding you together like glue. And those are just enzymes and structural proteins. Those are just two kinds of proteins that make you tick. So like, it's enough. It's enough that DNA is just a code for protein. Questions so far? God, second period, don't let this go to your head, but you guys, as classes go, are pretty cool. I don't know. I just like I can tell all of you are like actually with me and paying attention and stuff, and that doesn't always happen. So I appreciate it. Hand in the camera if you need more time. Yep, you got it. Hallie, you asked me a question. I can't remember what it was. You emailed me something.
It was about my shapes, like the cutout
It
for
was about
like
the shapes.
I think it was for part B.
Okay. Remind me at the end of class and we'll talk.
Okay.
All right. So we are now going to dive into transcription in some detail. So there will be some drawing, kind of like there was with replication, but not as much. And then we'll model it with paper shapes. Won't take too long, I promise. We're losing Evan, I think. He's yawning. And Dawson might be sleeping with his eyes open. I can't tell. You kind of have a weird light shining in your eyes. Makes you look a little bit like a cat. Okay. Gene, expression in detail, starting with transcription. You remember in replication that you needed one enzyme to unzip the two strands. And then you needed another enzyme on both strands, actually adding the new bases. That at least is simpler in this than it was in replication because we only have one enzyme working in transcription. The enzyme in transcription that does both of those jobs is RNA polymerase. Not to be confused with DNA polymerase, which was the one that did one of the two jobs in replication. RNA polymerase does both jobs. It unzips the DNA because there's no helicase this time. Remember, helicase was the enzymes that unzipped it in the other one. It unzips it at the front. It adds its own complementary RNA bases to one of the two DNA strands, not both. Remember, we're looking for a single strand of mRNA. And then it actually re-zips the DNA when it's done. Shuts the door on its way out. So now we're going to draw some pictures. What you've got here is that same DNA strand, okay, double-stranded, but we're showing the RNA polymerase already having unzipped a pocket of it and already having added some complementary RNA bases to the top strand. That would be called the coding strand of DNA. The one that's not being used is called the non-coding strand. Notice we're matching bases just like we're used to, except for that exception that RNA has, where wherever there would be a T if it was DNA, there's a U for uracil instead of thymine. All right. So I said it unzips the DNA at the front, it rezips it at the back, and remember that RNA isn't as sticky as DNA. It's still going to hold together in the single strand that it is, but it's not going to stick to the DNA as well as other DNA does. And so it's kind of just going to fall off. And what you wind up with is the DNA just like it was, zipped back up. But now you've got a separate strand, which is what we call the primary mRNA transcript.
Mr. Sandro?
Yes, sir.
Where's the test supposed to be tomorrow? Like
just in grade camp.
Okay.
Grade cam like usual. Yeah, no Google Meet, just be there at 11 and it'll open up for you. We're trying to get, I know it's probably frustrating on your end because I know you can't see tests that are coming until they drop, which we talked to grade cam programmers on our teacher workday Friday and told them that's something we want. And so they said they're working on it. But yeah, it'll drop in at 11. I've already got it set up. If it doesn't, please email me and I'll fix it. But I'm sure I checked the box properly this time.
Mr. Sandro, will you, I know it's at 11, but will you end up posting something on Google Classroom on Friday about it?
I'll probably honestly post it like this afternoon. But yeah, I guess I could maybe, I'll just set an auto post thing to drop in at maybe 8 a.m. to remind you tomorrow. I can do that.
Okay, cool. Thank you.
You're welcome. Okay, hand on the camera if you need more time. Cool. So paper shapes now, and I'm going to do this along with you. I brought some home. I have all of the ones that you would need. You might be short a few, so I'm going to do it on camera so that you see what it would look like if you're missing pieces. So I'm going to turn my camera down here onto my desk. And so we're going to recreate what we did yesterday, which is build a left-hand DNA strand that's top to bottom, TTA, TCA. It's going to be upside down to you because I have limits, and if I tried to do this upside down, I would mess it up. Okay. T, T, A, T, C, A. And then the other strand, which of course is going to be upside down. This is going to still be all DNA. We're still just working with DNA. Or it's going to be A, T, A, G, T. So I'll wait a minute, let you catch up. And so remember, we are modeling transcription here, not replication. So what's going to happen is, like in replication, we're going to move the two strands apart. And that's the job of the RNA polymerase in this case, not the helicase. But then instead of adding DNA to both old strands, we're going to add RNA just to the right-hand strand. And so you're just going to put the ones in that fit. Uh-oh, what did I do? All right. Oh, I took one of the DNAs. I will say, I get kind of impatient in class when everybody's not building as fast as I want them to. This has gotten me over that real quick. Having to do this myself for the first time in a long time, it's hard. I feel your pain. That one goes there, and that one. Yeah.
Mr. Sandro,
Yes, John.
on the right side where it starts as AA, I don't have 5 A's. I only have 4.
You're saying you don't have enough DNA.
Yeah.
And that is perfectly okay. Just kind of pretend it's there. What's really important to us and what you do have enough for, or what you should have enough for, if I looked at the sheets right, is the RNA. So that's the strand I just built in the middle there. That's going to be what we work with from here on out. So as long as you build that correctly, which I'll go ahead and put that in too, okay, that's your top to bottom, that's your RNA strand. Make sure that's correct and you're good. Any other questions so far? So kind of in the simplest sense, that's transcription. I'm going to take you the rest of the way through with the paper shapes so that you know what you're doing when you get to the assignment for today. And what we're doing with the paper shapes in exactly this order is exactly what you're going to do for homework. Okay, you'll be answering questions along the way, but that second paper shape worksheet has you building exactly what I'm building here. What we can do now that we've built the messenger RNA strand is we can get rid of the DNA. We can move that aside. Remember, in real life, it just zips back up like nothing happened. And so now we have a strip of messenger RNA that in real life is going to go squirt out of the nuclear pores into the cytoplasm and go find the ribosome. Now, I don't have a paper ribosome, so you'll just have to imagine the big glob that looks like a sandwich, and it kind of clamps down on the messenger RNA and holds it in the right spot for the tRNA to come in. Now, this is where the stuff I had you cut apart the other day comes in. Actually, that was yesterday, which is the tRNA molecules, these big guys here. This is where you want to get them and the amino acids out. You should have 2 of each, 2 different tRNA blocks and 2 amino acids, which you can tell are different, even if they look the same at first. One has 2 tabs, here and here, and one just has the 1 tab. So we're going to do this in the order it happens in the cell. We've got our messenger RNA here. What happens next, before the tRNA comes in and mates up with it, is the tRNA grabs the correct amino acid. And so notice that this amino acid doesn't have the right number of tabs. Sorry if we're talking like kindergartners here, but like this one does, right? That one fits. This one doesn't. So it grabs this one. Yeah. And then this tRNA grabs this one because it has the 2 slots instead of just the 1. So tRNA's job is to go out in the cytoplasm, grab the right amino acids, because remember, amino acids are what proteins are made of, And then it comes in and 3 bases at a time matches up with messenger RNA and brings the right amino acid in and hooks the amino acids together. And what just happened right there, that connecting to that is the whole point of this. The whole point of this is to bond amino acids together. You get one that's like 100 to 500 of these things long, and it folds upright, and now you've got a protein. So that's the process. That's transcription and translation, right? Transcription was what I did with the DNA and the RNA. Translation is what just happened here. Questions at this point? All right, good job sticking with me. The good news is that your homework, at least the paper shape instructions, are going to follow exactly what I just did. So if you need to, I am going to post this video online for today. If you get in the middle of it and you get lost and you want to look at what I did, that's fine, do that, okay? But the second worksheet, front and back, I'm going to post digitally today, is going to be due along with an uploaded picture of the whole assembly. What I ended up with here, this thing here, you need to have a photo of that because I know you have all the pieces to do that. And that's going to be your paper shape grade along with the worksheet. So did I drop it in already? I did, it's in there. It looks like this. Paper shape worksheet number 2. So you're going to complete that, which looks like this, and then you're going to, third page isn't anything, and then you're going to upload a photo of your whole tRNA, mRNA, amino acid assembly, and we're going to call that good as far as what's due tomorrow. Where did you go? There you are. Questions? Test point recovery is tomorrow from 11 to noon. Homework worksheet and photo upload is due at 2:30 tomorrow. I may post a brief video or something tomorrow, but no guarantees. If I do, it'll be by 11 a.m. And that, if you don't have any further questions, is class for today. If you want to ask a question on a smaller scale, stick around. If not, have a great day and a great weekend.
Mr. Sandro,
Gavin.
did you say that the test was on Google Meet or was not?
No,
Okay.
just grade cam like we usually do. Well, yeah, you've been remote before. You've done that, right?
Yeah.
Okay, good.
All right, thank you.
You are welcome.
Mr. Sandro,
Yes, Sydney.
I won't be able to do the 11 to noon because I will be driving to Tennessee.
Okay. What you'll need to do is have your mom or dad contact attendance, right? Just have them call attendance just like you would for a regular absence. And as long as that's an excused absence, we can set up another time. When do you want to do it?
I can do it Sunday.
Sunday?
Or, yeah.
Yeah, I can set it for Sunday. What time?
Anytime. I'll be back Saturday night.
You want to just keep it 11 to noon Sunday?
Sure. Yeah.
Easy to remember. 11 to noon Sunday, Sydney. Got you.
Okay, thank you.
You're welcome. Carissa.
Hey, Mr.
Okay.
Sandro.
Hey, what is up, man?
Not much.
I wondered who that was lurking in the background. I should have known it was you.
Yeah, I was looking at our notes and remembered all the fond memories of this class.
Like repressed trauma, PTSD.
Nah, like this is the one class I studied a lot for, but I always got like 100% to like 90 to 100% on the test.
Yeah, you were a beast, that's for sure.
My computer's about to die.
Yeah. Is she doing good or not so much?
She's doing great. I don't know. She's not one of the ones that
That's good. There's
she's
no
not
way
one to worry about it all. What's that?
There's no way she's doing better than me. She said that
Well,
she is.
you set a high standard, but she's got her own strengths.
Yeah. Oh, you have a good day.
You too, man.